The overall goal of this technique is to measure how nematodes and their neurons respond to external mechanical stimuli using a microfluidic chip setup with pressure actuators.
This method can answer key questions in the fields of mechanobiology and sensory biology, such as how cells, tissues, and animals respond to mechanical stimulation.
The main advantage of this technique is that it sufficiently reduces the worm's mobility in a non-invasive way to allow high-resolution imaging while still having access to the worm's cuticle for mechanical stimulation.
This method can also be used to study the influence of mechanical cues on development. It could even be applied to other systems such as ex vivo organ explants or other animals of similar size to C. elegans.
Set up a microscope system for simultaneous excitation of GCamp and RFP. One option is a discrete light source that transmits only cyan and yellow light. For viewing, use a 10 times objective and a high magnification objective. Also, send the image to a computer via a digital camera. Next, include a fluorescence cube and, if needed, an excitation filter. Add a dichroic mirror to the cube that reflects cyan and yellow light and transmits green and red light. Set up a beam splitter with a long-pass dichroic mirror with a cutoff at 570 nanometers. Also, provide a bandpass emission filter for green light at 525 nanometers with a 50 nanometer width and a bandpass emission filter for red light at 632 nanometers with a 60 nanometer width. Project both beams into the camera's field of view. Make sure the green part is projected to the top half of the screen and the red part to the bottom.